Evening, folks. And tonight I am going to be talking about, um, it's kind of like a review. I'm gonna do half of the mitosis worksheet, and then the other half I'll be doing in another video. I tried to do one full video of explaining all of the learning objectives from the mitosis worksheet, but apparently according to YouTube it was so long, so I'm gonna split it up into two different chunks and hope that it lets me upload. Okay, and so let's take a look. Okay, so first let's get into kind of why the learning objectives are really important. So the learning objectives, I feel like in class we don't really touch on them, but obviously it's expected to know these things, right? For mastery, complete mastery of the content that's presented on the exams, right? And so let's take a little peek at some of these answers for the learning objectives. Okay, so the first section is talking about the cell cycle. Describe the purpose of cell division. This one's pretty self-explanatory, but obviously we have old cells, old cells, dead cells in our body, and we need to make new ones to replace those old and dead cells. That's cell division. Describe the events of each of the stages of the cell cycle: G1, S, G2, M, and G0. Okay, it's something important to note is that M stands for mitosis, right? And that is completely separate from all of the other things discussed in this question. So you have G1, S, G2, and G0, which are all part of interphase, right? So interphase comes before mitosis, and there's no cell division, okay? So within G1, you're going to have cell growth, maybe some organelle replication. In S, the S phase, this is really important. This is when DNA is replicated. M is mitosis. G2, that's more cell preparation, kind of for, cell division, or M. And then G0 is where cells get sent if they are not supposed to divide. And keep in mind, G0 is typically depicted as kind of like this continuing cycle around interphase, because once cells, are sentenced there, they don't go to M, right? So there's no kind of change. Compare and contrast interphase with mitosis. I believe I mentioned this a couple of seconds before, but interphase is where there's no cell division. Mitosis, there is cell division. Okay, that gets us on to our next learning objective section, mitosis. Describe why DNA must be synthesized before mitosis. Okay, so if we think about the ultimate goal of mitosis, the goal is to create two diploid daughter cells, right? And so we want the same exact percentage of genetic information as our parent cells have, right? And so in order to get that same exact percentage, you're going to need more DNA to distribute naturally, right? So that each daughter cell gets a complete copy of the genome. Otherwise, you could end up with some weird, funky cells. Describe the anatomy of chromosomes and why chromosomes become condensed prior to mitosis. Okay, so if we think back to our prior unit, right, that was all about DNA. Our DNA as eukaryotes is very long and linear, right? I think it said in the book it was like 2 meters or something. So very, very long, right? And so we need some way to condense that DNA so that it's able to, get distributed in a normal way. And so that's what chromosomes are really, really useful for. So if we're looking at a chromosome, right, we're going to have our P ends, right? Or maybe that's more appropriate. P ends And then our longer kind of ends are our Q ends, something that's connecting them in the middle. That's going to be a centromere, right? Okay, define homologous chromosomes, maternal and paternal chromosomes, centromere, sister chromatids, gametes, and somatic cells. Okay, well, let's start with the easy stuff. Gametes are not produced by mitosis, but they're produced by a separate process called meiosis. Gametes are specialized cells. They're sex cells or germ cells, so they're used in the reproductive process, right? You have spermatocytes in males and oocytes in females, okay? Somatic cells are going to be just your normal body cells that are produced through mitosis, okay? Centromeres are going to be as I mentioned in our anatomy of the chromosome, they're going to be this thing that connects the two, the two chromosomes in the middle. Okay, so maternal and paternal chromosomes, for all intensive purposes, there's not, as depicted in this worksheet, there's really not much of a difference, right? But they're very important for meiosis. What you should know is that there is, that a zygote gets, both maternal and paternal DNA, right? Homologous chromosomes, those are going to be like our pairs of chromosomes, and those are going to be separated in meiosis, whereas sister chromatids get separated in mitosis, right? So if we think about this, mitosis, sister chromatids separate, meiosis, homologous chromosomes separate, at least in meiosis 1, okay? Define karyotypes and how they are used. Karyotypes are basically a tool for chromosomal mapping, right? So it gives you this kind of solid picture of an organism's chromosomes and the layout of those, right? Okay. So how they are used. Karyotypes are really useful for noting chromosomal abnormalities, right? Especially, you can note when there's been some unusual things. So trisomy 21, I believe, that's where you have an extra 21st chromosome, or better known as Down syndrome. You can see that very clearly on a karyotype, because as opposed to 2, you get 3 on that 21st chromosome. Okay. Describe the events that occur of each stage of mitosis and cytokinesis. Okay, for all intensive purposes, this can be, Dr. Apasami's PowerPoint is very clear about this, so that's an excellent resource. If you want a kind of clear picture of it, though, I recommend looking on Khan Academy. Sal from Khan Academy does a really great visualization of kind of the events that occur at these stages. Define whether sister chromatids or homologous chromosomes are separated in mitotic anaphase. I believe I just discussed this, but sister chromatids are going to separate during mitosis, right? So anaphase is when the chromatids are pulled apart to the opposite ends. Describe the roles of the mitotic spindles, kinetochore, centrioles, aster, motor proteins in separating chromosomes in anaphase. Okay, let's start from the beginning. Mitotic spindles are what are going to extend from the centrosome, okay? And the centrosomes are going to be, I believe they're proteins, but they're these structures located at kind of like the tops of the cells. The mitotic spindles are going to extend, attach, and pull the chromatids apart. Okay. Kinetochores are the proteins that are actually going to attach to the mitotic spindles. They're located on the chromatids. they're going to pull them apart to the opposite ends. The centrosome, I just touched on this, but there are these structures located kind of at the tops of the cells. They'll attach. They'll be attached to the mitotic spindles. Asters. So asters are these special protein structures also located at the tops of the cells. They work to elongate. They're composed of like this kind of like blob and then these smaller microfilaments that will work to push the cell, to widen the cell, right? And then that leaves you with motor proteins. Motor proteins kind of help to push the chromatids apart to opposite ends of the cell. Describe the role of the contractile ring of actin-myosin in cytokinesis. Okay, so cytokinesis, right? That is the division of the cytoplasm. Okay, so figure in telophase, we have one big chunky cell. Cytokinesis is going to, create this division of the cytoplasm, which will result in the two daughter cells. So within animal cells, we have what's called a cleavage furrow, and what that basically means is that actin and myosin are just going to work to pinch off this giant cell to create two daughter cells. Describe the role of the cell plate in plant cells during cytokinesis. So the cell plate does basically the same exact thing. It's just going to create kind of this protrusion that's going to create two plant cells. Describe how bacteria reproduce. Okay, so bacteria, unlike us, they have this loop of DNA, right? We have long and linear DNA. Bacteria are prokaryotes, they have this loop, so they can replicate their DNA more quickly than we can, and so as a result, they're able to kind of divide and create these two daughter cells really quickly. They replicate their chromosomes. They also have proteins that elongate their cells, so they'll have one big, long, chunky cell like we will have, and then they just fuse to create two daughter cells, and that's called binary fission. Okay, and that's asexual reproduction. Something also to note, mitosis is, it's found within organisms that sexually reproduce, but the cells that are being generated are not sex cells. That's meiosis. Okay, so that's just something to note. A challenge question for you. What you can draw a cell that is stuck in telophase. What would that cell look like if it failed to have cytokinesis happen? And so that's just part one of an explanation for the mitosis cell cycle learning objectives worksheet, and I'll be back with part two for you soon.